Hello, everybody, and welcome to today's webinar, Expanding the Cell Painting Toolkit, Optimizing Imaging Across Diverse and Challenging Cell Lines. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Dr. Andrew Sullivan, Principal Scientist, Immunology Concept Life Sciences. Dr. Sullivan, you may now begin your presentation.
Thank you for inviting me to speak today and show some data about some recent research that we've been conducting at Concept Life Sciences. I'd also like to say thank you to everyone who's joined this webinar, and I hope that you find what I have to present both interesting and hopefully useful for any work you might be planning in the future. So I'll be talking about cell painting and the challenges and modifications that can be applied to the workflows to really get the most out of the technique itself. I'll start with a brief introduction to myself. I currently work as a principal scientist at Concept Life Sciences, and I've got previous experience in biotech, large pharma and academia, as well as currently being part of a CRO. My background is originally in pharmacology and drug allergy, although I've since expanded to take in multiple areas of investigation across the field of immunology and immuno-oncology. I work extensively with different cell types and regularly find myself involved in bespoke assay design to answer difficult scientific questions. Just to give some information on ourselves as a company, Concept Life Sciences are a CRO based throughout the UK, although myself I'm located in Edinburgh, and we offer a wide range of drug discovery services throughout the development process, from initial target validation and hit identification all the way through to candidate selection and clinical application. We have our own chemistry, biology and ADME services, allowing a fully integrated offering for our clients' requirements. And we really pride ourselves on being able to work in an expert-to-expert communicative way to provide any potential requirements that our clients might have. So I'll begin with a brief refresher on what cell painting is, how it works, what it measures and why it's become such a powerful tool in phenotypic screening and drug discovery. Next, we'll explore why it's important to move beyond the standard cell lines. We'll look at how incorporating diverse and more physiologically relevant models can improve the biological insight that we gain from these assays. Now, of course, working with new or difficult cell types comes with challenges. I'll walk through some of the common issues like staining and adherence and share suggestions on alleviating them. We'll then dive into a few case studies, highlighting how we've successfully applied cell painting to iPSC-derived cells, primary cells like macrophages and other non-traditional models. These examples will show the real-world value of expanding the toolkit. And finally, we'll wrap up by looking at how this expanded approach enhances our ability to predict toxicity, uncover mechanisms of action, and even identify opportunities for drug repurposing. All of the work that I'll be showing you has been performed using the Thermo Scientific high-content screening platform that's shown here. It's an incredibly easy tool to use that allows the generation of massive data-rich experiments with excellent image resolution and data quantification. So what is cell painting? Well, it's a high-content imaging assay designed to capture rich morphological information from cells using multiplex fluorescent dyes. Each dye targets a specific organelle, such as the nucleus, the mitochondria, and the endoplasmic reticulum, and gives a comprehensive view of cell architecture. In once imaged, the assay allows us to extract thousands of quantitative features per cell, including size, shape, intensity, and co-localization. Now, this makes it incredibly powerful for detecting subtle phenotypic changes in response to drug treatment, even when those changes aren't necessarily visible to the naked eye. And it matters because it allows us to approach screening from a hypothesis-free, phenotypic first perspective. Instead of asking, does this compound inhibit target X, we're asking, how does this compound affect the cells? Now, this is especially valuable when we don't yet know the full biology of our target, or when we're screening for novel mechanism of actions. Compounds with shared mechanisms tend to induce similar morphological changes, and this lets us cluster compounds by phenotype and infer their mechanism of action from the data. Cell painting can flag early warning signs like mitochondrial fragmentation, and it's both cost-effective and scalable to large compound libraries. So historically, the field of cell painting has focused on a small set of well-characterized cell lines because they're robust, easy to handle, and readily available. Examples include the U2OSs, A549s, and to a lesser extent the HeLa cells. Now, these lines are consistent in both growth and morphology, meaning they provide a great technical foundation for large-scale screening and method development. However, this convenience comes with limitations. Firstly, they're all cancer-derived and often highly aneuploid, and that means that they're often genomically unstable and can have altered signaling pathways compared to regular physiology. A good example of this is the dampened p53 response seen in some cancer lines, which affects the interpretation of DNA damage signatures. Their tissue origin can be poorly representative of the biology that we're trying to model. For example, the osteosarcoma-derived U2OSs being used to study hepatotoxicity or neurodegeneration isn't ideal due to the lineage of the tissue. Now, I've highlighted a couple of cell lines that are completely different morphologically to the standard cell painting lines to showcase this. The B35s are a neuroblastoma cell line, and the SK-HEP1s are derived from liver adenocarcinoma. Even by a quick glance with the eye, you can tell that they're significantly different to the lines on the left. Now, to make cell painting a truly predictive and translational tool, we need to extend our panels to include a broader range of cell models. So the term itself has been around for less than a decade. It was first outlined by a Broad Institute paper in 2013, which was then expanded upon in the 2016 publication by Bray et al., which coined the term cell painting. Now, over the following years, there's been a steady increase in papers being published referencing the term, with over half of them coming in the past couple of years. It speaks to how much the technology has improved, but also the understanding of the importance that cell painting and the associated data that it generates can provide from an insight perspective. And currently, the vast majority of publications focus on a relatively small selection of cell lines. If you do a search for cell painting plus A549, you'll get about 70% of the total results that you would do with cell painting alone. It's understandable why this is the case. The initial papers that developed cell painting as a technique looked at these cell lines. The cells themselves are also easy to culture and maintain. As I'll describe later, there are a lot of challenges associated when you work with potentially non-ideal cell morphologies from a maintenance standpoint. Now, think about how a kinase inhibitor might reduce proliferation in a cancer cell, but could potentially induce differentiation in a neuronal precursor. Those phenotypes aren't interchangeable. By using different cell models, epithelial, hepatic, or neuronal, we can uncover how compounds can behave in the context of actual tissue biology. The cell type you use fundamentally changes the phenotype you observe in a cell painting assay, and this is due to a multitude of factors: gene expression, receptor availability, metabolic state, and organelle content all vary widely between different cell types. As an example, a dopamine transport inhibitor will have little effect in epithelial cells, but could significantly alter dopaminergic neurons. And similarly, a compound inducing lipid accumulation may produce a strong change in hepatocytes, but not in fibroblasts. Put simply, the same compound can yield different phenotypic signatures depending on the cellular context. And this cell type specificity is a major opportunity. By profiling compounds across different models, we can reveal context-dependent mechanisms of action, uncover tissue-specific toxicities, and build more detailed and complete pharmacological maps. By considering the biological questions and selecting cell lines based on them, we can increase the translational power of cell painting. Now, this is great in theory, but expanding into more physiologically relevant models introduces more complexity. Suspension cell lines can prove to be tricky to image just due to the nature of their growth. You have to adhere them to a plate using a substrate, which can bring its own imaging challenges, and then you'll have the typically small round nature of the cells proving hard to focus on an image. And similarly, primary cells can prove challenging as well. These more closely represent actual tissue biology, but are variable and limited in supply. Imaging them is harder. They may not adhere well or even at all. They may grow slowly, and their morphology can be heterogeneous. Now, iPSC-derived cells are another option. We can differentiate iPSCs into multiple cell types, such as cardiomyocytes, cortical neurons, and microglia. These models are becoming more reproducible and scalable, and they carry with them potentially disease-relevant genotypes. iPSC-derived microglia can help screen for neuroinflammatory effects of CNS targeting compounds, which lines like the U2OSs would miss entirely. They do bring a lot of challenges themselves, however, such as requirements for bespoke media, careful plate coatings, and advanced segmentation strategies for accurate image analysis. All of these additional models can help us close the gap between in vitro assays and in vivo human biology by enabling better predictions of drug efficacy, off-target effects, and tissue-specific toxicities. Now, what does a typical cell painting workflow look like? On day 0, it typically involves plate coating if it's required and cell seeding. You should aim to achieve a monolayer of well-spread cells with minimal clustering or overlapping, as cell density is critical. Too sparse and you'll have trouble generating data in imaging, and too dense, segmentation becomes practically impossible. A lot of different cell types also will not grow or even die off if they're seeded suboptimally. On day 1 after acclimatization and resting, the compounds or the tool agents are added. Depending on the desired phenotype, for example acute stress or chronic adaptation, incubation times can vary between hours to days. And then at the final time point, it's time for staining and fixation. Now, the standard cell painting panel consists of 6 fluorescent dyes targeting key organelles, as shown in the table, with a combined cell painting kit available for the complete plates, such as the Imagit kit that Thermo Fisher offer, and I've used extensively. Now, put together, this workflow forms the backbone of most cell painting experiments. So here's a few tips I can offer to help you get the most out of your experiments. Now, most protocols aim for between 60 and 80% confluency at the time of imaging, and a consideration should be made of the cell morphology prior to plating to achieve the optimal density, as it will be different depending on the cell type being investigated. Different cell types exhibit different morphologies, adhesion requirements, and proliferation rates, which will all affect how they behave in a plate. Immortalized lines like U2OSs and A549s are straightforward. They'll adhere well and spread consistently. However, in contrast, primary hepatocytes or iPSC-derived neurons often need specific extracellular matrix coatings to successfully adhere. Now, coating reagents can also autofluoresce or potentially interfere with dye binding. Testing for background signal in each imaging channel during assay development is essential, especially in highly complex assays like cell painting. And then finally, gentle washing steps are important for non or potentially loosely attached and adherent cells. Using automated liquid handlers with slow aspiration rates and tilted plates can help minimize any cell loss or lifting that may occur. As I've mentioned, cell painting relies on multiplex fluorescent imaging. While this is powerful, it introduces a key technical issue, which is spectral overlap or channel crosstalk. Many fluorophores have broad excitation and emission spectra, and adjacent channels can bleed into each other. For instance, some cyto dyes can emit strongly in both green and orange ranges, overlapping with the CON-A label. And this is shown in the comparable fluorophore for both CON-A and cyto14 in the original cell painting panel is Alexafluor 488. And if not corrected, this overlap can alter phenotypic signatures. A high mitochondrial signal may appear as increased actin or endoplasmic reticulum texture, leading to misinterpretation. Now, the image at the bottom left shows both nucleoli, endoplasmic reticulum, and plasma membrane staining using the standard cell painting panel. While differential staining can be observed, signal can be detected in both channels that makes identifying specific organelles difficult. Swapping the endoplasmic reticulum stain to a different fluorophore, in this case Alexafluor 750, as shown in the right, significantly reduces the signal spillover and allows differentiation of the distinct organelles. Now, here's a couple more tips we've been able to identify and could prove useful if you encounter any issues during your setups or optimization. First, dropping the concentration of mitre tracker can help to reduce cytotoxicity in certain sensitive cell types. However, this will come at the trade-off of potentially poorer or weaker staining. Additionally, as any scientist knows, having known controls are the backbone of any experiment, and it's no different for cell painting. Some good reference compounds are highlighted here. And earlier I mentioned that growth morphology can make imaging difficult. For instance, the HepG2 cell line loves to grow in clusters and on top of one another. Now, this makes for a very happy group of cells, but a very tricky thing to image. And to counter this problem, you can employ some Z stacking protocols. This is where you align the Z plane where you would image the monolayer as normal, and then instruct the software to also image a set amount of microns both above and below that location to build up a composite image that can be tens of microns thick. The software will then perform compression on all of these individual images, and the result is a 2D interpretation of a 3D picture. I've shown an example of this with the HepG2 cells below, showing that even though they don't grow in a single monolayer, it's still possible to get a nice monolayer equivalent image from them. Now, extracted features from the images form the foundation of all downstream analysis. As much as everyone loves a nice picture, the analysis and the underlying numbers that are generated are what makes cell painting such a powerful tool. After staining and imaging, each cell is segmented computationally into various compartments, typically defined through the nuclei channel for single cell identification. The images shown here are taken directly from an analysis performed on the HCS Studio software, which is what drives the analysis on the Thermo Fisher instrument. Feature extraction means the quantifying aspects of each cell's morphology and signal distribution, and these can include intensity, which is how bright the staining is in each compartment or channel, texture, the granularity or smoothness of different signal patterns, shape, which is combined measurements of area, perimeter, eccentricity and compactness, and spatial features, which describe the relative positioning of different cellular components. All these values are then exported as numerical values in a sometimes very large spreadsheet, which allows downstream subsequent analysis to be performed. Channel level granularity of feature extraction is where the phenotypic differences between treatment groups can become evident. Labeling key organelles allows individual cell-to-cell comparison and gives an indication of the phenotype. Nuclear shape and DNA content informs on cell cycle phase, nuclear fragmentation or multinucleation. Endoplasmic reticulum stress and expansion can manifest as an increased texture. The cytoskeleton is highly dynamic and responds to various toxins, while assessment of vesicle density, compactness and overall Golgi dispersal gives an indication of health. Each of these readouts contributes orthogonal information. When combined across channels, they can describe complex phenotypes such as cellular senescence, immune activation, or neurotoxicity. So, using all this information, what can we do to expand the cell painting toolkit? Well, there are a couple of options available, so to investigate them further, we established 3 case studies at Concept to evaluate their potential, looking at the areas that have been previously discussed as challenging. First, and theoretically the easiest, is expanding the cell painting line library to encompass a wider panel of tool cell lines, and this should allow better reflection of morphology and a greater array of tissue-specific responses to be assessed. To start with, I'll present a case study where we applied cell painting across 12 distinct cell lines. Now, these were chosen to reflect a range of tissues, lineages and oncogenic states. Our goal was to capture a broad phenotypic spectrum and assess how baseline morphology and compound responses varied by cell type. Lines like RT4, HepG2, PC3 and 22RV1s represent diverse carcinoma-derived cell lines. These originate from tissues such as cervix, lung, bladder, pancreas and prostate, and they exhibit notable differences in overall phenotype, organelle distribution and function, all important features for morphological profiling. Lines like MRC5, which is a human lung fibroblast line, are commonly used in cytotoxicity testing. This provides a comparison to the carcinoma-derived lines as it's not considered cancerous. HMC3 is a human microglial cell line and informs on CNS-relevant morphology, while the osteosarcoma U2OS is well characterized in cell painting as mentioned previously, and it's one of the model cell lines to compare the others against. Now, from an assay development perspective, we observed that the growth characteristics, tolerance to seeding density, and even simple things like amenability to imaging or monolayer growth varied significantly between cell lines, and this influenced both imaging and feature extraction. As always, it's important to be able to assess the baseline phenotype. Any assay being performed requires robust controls. And for that reason, having both a media and vehicle condition was essential to be able to have an unperturbed phenotype. It was also an important consideration, as if imaging the baseline phenotype proved difficult, it would inform that there would probably be significant modifications required to image other conditions, or even that the cells themselves just didn't fit with the cell painting procedure. The compounds chosen all have known mechanisms of action, which are distinct from one another. Different MOAs should result in different phenotypes, and testing both low and high concentrations would allow the investigation of potential susceptibility to certain compounds by certain cell types. So after running the experiment, imaging the plates and looking at the generated data, what did we find? Well, thankfully that we were able to image our new cell line panel. Comparing one of the model cell lines, in this case U2OSs, against some of the newest lines that we had chosen to investigate, HUVEX and MRC5s, showed that it's possible to distinguish a clear phenotype of the different cells. MRC5s in particular are something I would consider to be excellent for cell painting, as repeated imaging with this fibroblast line gives consistent results. Clear delineation of the nucleoli and individual actin filaments, as well as being able to distinguish individual mitochondria, mean that these are perfect for this application. HUVEX, on the other hand, proved to be more challenging. Anyone who's used them before will know that they're particularly difficult to work with and put down large amounts of debris during their growth. As such, the images can appear dirty due to fragments causing autofluorescence. Even with this, however, you can still pick up the cellular structures and organelles. So we're able to generate images, but can we actually determine anything from them? And as I've mentioned, there's a vast amount of data that comes along with cell painting, so much so that it's unwieldy for one person to analyse and practically impossible to do without help from algorithms. To visualise all this data, we can employ a dimensionality reduction technique such as principal component analysis. This technique crunches all the numbers and projects it into principal components or groups that capture the greatest variance in the dataset. It's an incredibly complex technique, and I'm not a statistician, so I won't go into it in detail, but suffice to say it allows distinguishing between different phenotypes where clusters that appear close to each other on the plots should look similar. As an example, the PC3 cells that we used for part of this experiment were very difficult to get a clear image of. They're small, rounded, and they just don't image well. However, the imaging analysis was still able to assess the different phenotypes that were being stained and perform an assessment. The plot itself looked like this, but did that match up with the different phenotypes? And when you compare the representative images, they're clearly different to one another. And what's encouraging to see is a dendrogram that was generated alongside that PCA plot showed similar results. In the group containing high concentrations of compounds like fenbendazole, vincristine and JNK inhibitor were all clustered together and were all shown in the images to strongly alter the phenotype of the cells towards apoptosis. I'll also show some data for HeLa cells. Now, thankfully, these are much more amenable to the generation of clear, crisp images. Following the PCA analysis, they broadly split into 3 groups: healthy, altered, and really very unhappy. Importantly, looking at the images themselves allows us to subject them to a simple eye test. Is what the images show backed up by the underlying data? And then, interestingly, in the dendrogram analysis, the treatment groups and their relation to each other have shifted around. Now the low concentration of fenbendazole and JNK inhibitor are distinct from the control groups, whereas in the previous slide they were much closer together on the dendrogram. This would suggest that in this instance, those compounds are having a more significant effect on the cells in this case, compared to the PC3s that were challenged with the same concentrations. And this is where having that diverse panel of cell types to assess can be crucial for drug discovery. One cell line will not respond the same as another, and it's borne out in the images, the raw data, and the analysed data. Now, does what I've just mentioned become detectable in the complete data set? To do that, we took all of the raw data generated from across all the cell lines and combined it together. In this example, we looked at the control groups alongside both the low and high dose of fenbendazole alone to assess if a different phenotype could be picked up based on the cell type. And following PCA analysis, 3 distinct groups became apparent. A relatively normal control group, which contained the majority of both the vehicle and media treated wells. A second group distinct from the first, and upon examination this contained cells that had unique phenotypes, such as multinucleation observed in UTOSs and certain instances of blebbing at the ends of actin filaments in cells like the T24s and the MRC5s. Now, curiously, there was also a couple of vehicle treated wells here, although with such a large panel of diverse cell morphologies, some overlap is to be expected. And the final group was clearly dead or dying cells, and giant vacuolar structures or cell fragmentation was common here. And then a further look at the grouping showed even more information. Highlighted in green, you can see a cluster of purple wells, and these correspond to the high concentration of fenbendazole, indicating that in these cells nothing significantly different from the control wells was being observed. And this again highlights that the same compound can have different responses. In the highlighted blue sections, pink wells, which is a low fenbendazole concentration, have apparently either a significant or non-significant response depending on the cell type. And this gives an indication that some of the cells tested are highly susceptible to fenbendazole, even at low concentrations. So as a summary, cell painting is traditionally focused on a small number of well-characterized, easy-to-image cell lines. However, the technique is not inherently limited to these models. The established protocol can be applied across a wide variety of cell lines, although keeping the established cell lines in any screening panel would be optimal. One of the strengths of the approach is the ability to analyze multiple cell lines in parallel. Each line brings its own unique morphological signature, and by profiling them together, we can capture a richer, more nuanced version of compound effects. By using this diverse panel of cell lines, it allows us to detect lineage-specific phenotypic responses that might only occur in certain cell types. And these responses can provide critical insights into a compound's mechanism of action or potential off-target effects. This is especially useful in comparative studies where understanding how different cell types respond to the same challenges or identification of cell type specific vulnerabilities is critical. Some cell lines are often excluded from high content imaging due to challenges like poor adherence, irregular morphology, or low contrast. However, with optimized staining protocols, imaging parameters, and analysis pipelines, we've been able to extract meaningful data even from these difficult models. Including these cell types expands the biological relevance of our assays and helps ensure we're not overlooking important phenotypes simply because their cells are harder to work with. Now, moving to more physiologically relevant cells is key for the future of drug discovery as a whole. iPSC-derived cells are moving to the forefront of research due to their pluripotency and potential applications, but can they be used in cell painting? At this point, I'd like to introduce our collaborators, who have been involved in this case study investigating the role of iPSCs in potential cell painting applications. Cytochrome will work to provide a wide range of iPSC-derived cell types from genetically diverse donors, allowing the formation of multicellular models of health and disease. They specialise in hepatic and cardiac iPS-derived cell types and aim to improve inclusivity in drug development to better represent the global population. Ignota aim to rescue and repurpose promising drug candidates that have unfortunately failed elsewhere. Through the application of their AI modelling, they combine both bio and cheminformatic datasets to investigate and solve drug safety issues. Through the prediction of biological effects via deep learning, they aim to reduce the potential for toxicity without sacrificing therapeutic effectiveness. iPSCs have immense potential in the future of phenotypic screening, largely due to their capacity to differentiate into almost any cell lineage. This gives them a patient-tailored approach that is becoming more common in personalised medicine. However, these highly specialised cells do come with their own set of challenges. iPSCs are highly sensitive to physical and chemical alterations, minor changes in media composition, different stiffness in growth substrates, or even passaging techniques can trigger differentiation or cell death. These cultures can be prone to the formation of growth colonies, with spontaneous differentiation a risk at various points in the culturing process, such as overconfluency or undernutrition. And this variability affects fluorescent imaging resolution and signal quantification, especially for assays that require uniform markers. In the data I'll show in the coming slides, there was significant differences in growth kinetics of the different iPSC-derived hepatocytes, highlighting this challenge. And then finally, the cells themselves are both time and laser intensive to prepare and maintain. Initially, cell painting was designed to be as easily accessible and as cheap as possible to get the most out of the data from the least amount of input. And this is where the transformation from the beginnings of cell painting to more diverse, disease-relevant alternatives becomes apparent, with the increase in complexity and cost being mirrored in the increase in valuable information that can be extracted from the data. The first question that needed to be answered in this instance was, can we even use cell painting as a way of assessing iPSC-derived material? It's not something we'd done before, so there was an element of uncertainty about what might be observed. As I've just mentioned, iPSCs can have different growth kinetics and morphologies, so there was no guarantee that the cells would be amenable to imaging after the cell painting protocol. There was also a risk of significant autofluorescence, as we see with long-lived cell lines or material that likes to put down additional matrix such as UVEX. However, I'm pleased to say that the imaging was able to be performed and some quite lovely images and subsequent data sets were able to be generated. Along with the black and white individual channel images, the pseudo-colour composite image at the bottom highlights an example well that was captured with minimal modifications, showing that these iPSCs can be used with this protocol. The team at Cytochroma wanted to investigate their iPSC-derived hepatocytes and settled upon paracetamol as a perfect tool compound to assess if the cell painting protocol was fit for purpose. Now, paracetamol is incredibly well characterized, with high doses resulting in an overwhelming of the detoxification pathway through the liver, leading to the formation of a highly reactive intermediate compound that can cause severe hepatotoxicity. Because of this, in theory, coming in with increasing concentrations of paracetamol should result in a more cytotoxic response in these iPSC-derived hepatocytes. And through the analysis, we were able to pull apart the treatment groups into 4 distinct clusters, which confirmed our hypothesis. The highest 50 millimolar concentration of paracetamol was heavily toxic and distinguishes as a group in its own right. The next group of 10 to 20 millimolar again separated out, showing an altered phenotype compared to both control and the highest doses. When the lower concentrations of 2 and 5 millimolar also cluster together, with the lowest doses in the vehicle forming the final cluster. And this is confirmed upon investigation of the dendrogram, where the 50 millimolar dose is the furthest apart from everything else, owing to its clear toxic effects. Now, following that, the team at Ignota were investigating a compound that's also implicated in hepatotoxicity, so wished to use these iPSC-derived hepatocytes as a model to investigate the phenotypic response of increasing concentrations of the compound. After performing the analysis, the clustering algorithm was again able to pull apart different treatment groups. In this instance, staurosporin was included as a positive control to give a toxic or a dead phenotype, with the staurosporin treated wells giving a unique phenotypic profile that clustered together, distinct from any of the other groups. Similar to the paracetamol data previously, a dose response of the compound and its phenotypic effect was able to be pulled apart in the data. Higher concentrations gave a more distinct and different phenotype, which clustered together and far apart from the vehicle. Lower concentrations, up to about 10 micromolar, showed an altered phenotype that sat somewhere in the middle, with a low dose of the compound having no significantly different effect than vehicle alone. A quick glance of both the vehicle and the 30 micromolar treatment groups does not immediately show a significant difference in the pictures, at least to my eye, and especially not compared to the massively different profile seen in the staurosporin images. However, the analysis pulled the vehicle and the 30 micromolar groups apart significantly, speaking to the power that cell painting and the analysis has in detecting subtle morphological changes. Even though not immediately apparent to the eye, the underlying data is able to detect the difference in the phenotypes that suggests some sort of aberration to the cells indicative of a non-healthy state, highlighting the potential use of this tool in predictive phenotyping of novel compounds. The good thing about this study was that it wasn't done on a single iPSC-derived line, but on several. We have data from multiple different potential phenotypes to investigate the responses that were being generated following challenge. On this different line, again, performing the analysis allowed the clustering algorithm to pull apart different treatment groups. As we expect, staurosporin treatment resulted in a unique phenotypic profile that clustered together repeatedly and distinct from any of the other groups as we've previously seen. And again, a compound dose response was observed. This time there was less distinct grouping of the different treatment groups, though a trend was still present. Again, the highest concentration of the compound was shown to be significantly different from all the other concentrations, which were closer to the vehicle group. A confirmation of this result was observed when investigating the well images themselves, which backed up what we were seeing in both the raw data and the PCA plots. Vehicle, high compound concentration and staurosporin treatments all visually look different, which confirmed the PCA data. To summarise, iPSCs offer a renewable and scalable source of human-derived material. This is particularly valuable in phenotypic screening, where consistency and biological relevance are critical. These cells can be cultured and stained using established protocols, allowing researchers to use morphological profiling without needing to redesign their workflows. In these studies, iPSC-derived hepatocytes have shown clear dose-dependent morphological changes in response to compound treatment. This demonstrates that these cells are not only viable in cell painting assays, but also capable of producing quantifiable, reproducible phenotypic data. Such responses are essential for building predictive models and identifying subtle biological effects that might be missed in less responsive systems. And then finally, we've shown strong proof of concept data supporting the use of iPSC-derived cells in toxicity screening. When exposed to known toxicants, these cells exhibit expected phenotypic changes, validating their use in predictive toxicology. Now, primary material is always going to be held up as the most useful gold standard for any sort of investigative experiment. Again, the question is whether the cells themselves will be suitable to the cell painting procedure in order to give further tools to perform our analysis. At Concept, we have a well-defined in vitro model that establishes functionally polarized macrophages resembling pro-inflammatory and anti-inflammatory states, enabling the study of macrophage-driven phenotypes and drug responses. To do this, PBMCs are typically isolated from either fresh whole blood or buffy coats. We perform untouched monocyte isolations, and this results in a highly enriched population of CD14 positive monocytes. Then, once isolated, these are then seeded at a density that allows for cell spreading and monitored over up to 7 days. Inducing the differentiation of these cells is then critical to the formation of the macrophages that are subsequently used in our assays. M1-like macrophages appear more flattened, irregular, and egg-shaped in appearance, while M2-like macrophages are typically stretched and can contain significant amounts of intracellular vesicles. Real-time imaging can capture morphological changes over time, such as adherence at day 0, spreading and differentiation by day 3, and phenotypic divergence by day 6, as shown in the videos. An example of the fully polarized macrophages are shown in the bottom left, with the distinctive phenotypes of the different lineages clearly visible. These changes reflect cytoskeletal remodeling and organelle rearrangement, which therefore should be also detectable via phenotypic assessments such as cell painting. Extensive profiling of the macrophages differentiated from peripheral blood monocytes using the procedure outlined on the previous slide gave a wealth of data of the phenotype of these cells, confirming that the lineages that were being aimed for were accurately expressed. M1-like macrophages represent a pro-inflammatory phenotype, while in contrast the M2-like macrophages are considered anti-inflammatory. Our qPCR analysis confirms the expected transcriptional divergence between these phenotypes, and cytokine readouts reinforce the transcriptional data. Co-cultured T cells also revealed clear functional consequences of different macrophage phenotypes. Now, these two flavors of macrophages look different, have different roles in the immune system, and trigger different responses. All of these things suggest that they should be amenable to some sort of phenotypic profiling, such as cell painting. These features can provide a visual fingerprint that can complement molecular profiling and could be used for downstream predictive assays. To prepare our polarized macrophages, we isolated out some CD14 positive cells, then employed the differentiation and polarization procedure to skew towards either the M1-like or M2-like phenotype. And once these cells were sufficiently polarized, we then plated them out into a fresh imaging plate and allowed them to rest and adhere before challenging them with a selection of compounds and assessing their effects in the cell painting assay. 3 different donors we used, with each condition being performed in triplicate to give some technical robustness to the data. And the compounds that were tested were similar to the previous case studies in this webinar, in that they have a known mechanism of action and should result in distinct phenotypes that could be detected following phenotypic analysis. Now, the most important thing we wanted to take from this imaging was simple. We knew that live phase imaging of the cells could show their distinct phenotypes, but would that also be able to be picked up in the phenotypic analysis that happens during cell painting? And as a reminder, I'm showing the phase contrast images that were previously generated, and then they can be compared to the images that were captured on the high content imager using the cell painting protocol. It's clear to see that the structures of the different polarized macrophages are significantly different and match up nicely to those saw previously. There are globular, fried egg-like structures present in the M1-like cells, while the M2-like macrophages are elongated and spindly, like what was seen in the real-time imaging. We knew that we had different phenotypes after examination of the images, but the real question was whether the data would back up what we were seeing. To that end, all of the metrics from this experiment were plugged into the analysis software, and once again we were able to show that these distinct phenotypes were able to be separated in PCA plots. For both the M1-like and M2-like macrophages, a separate distinct cluster was observed that corresponded with the staurosporin treatment, which is to be expected. Where differences could be seen was in the other compound treatments against the different flavors of macrophages. On the top plot, the M1-like cells show both rotenone and JNK inhibitor clustering as their own groups, distinct from both each other and the control wells. You can also see this replicated in the lower PCA plot for the M2-like cells. However, in this case, the groupings are a lot more fluid and less well defined. There's a merging of both the control groups into the rotenone wells, with only the JNK inhibitor able to be clearly separated. And this gives us an indication that although these cells are the same lineage, they do exhibit different phenotypes in response to the same challenge. The final test was to see what the data looked like when it was all pooled together. We now wanted to assess the same compound effect on different versions of differentiated and polarized macrophages. And after analysis, an interesting pattern emerged. Again, staurosporin was a distinct phenotype able to be detected on its own, although this was able to be split between the M1-like and M2-like treatments. The healthy phenotypes of these cells look different, and so did the dead and dying ones. Whether this is just to do with relative differences in autofluorescence or cell fragmentation that was left behind in the wells can't be determined here. The most interesting thing to come out of the data was that JNK inhibitor treated M2-like cells and rotenone treated M1-like cells were together in their own cluster that was practically the same. A look at the images for these wells shows us why. A visual inspection of the cells show them looking very similar, small and rounded with increased autofluorescence. Again, this is showing us that the data is backed up by visual confirmation, and that distinct phenotypes can be identified and clustered together regardless of their source. One of the key advantages of using primary cells in cell painting is the ability to detect tissue-specific toxicity early in the drug discovery process. And this could potentially uncover adverse effects that might otherwise go unnoticed in immortalized cell lines. Different cell types often respond differently to the same compounds. By using macrophages that have been polarized differently, we can identify lineage-dependent responses. And this helps us understand tissue-specific efficacy or toxicity and supports the development of safer, more targeted therapies. Incorporating a variety of primary cells into cell painting assays allows us to build a more comprehensive picture of compound safety. Instead of relying on a single cell type, we can assess effects across multiple tissues and improve our ability to predict potential adverse outcomes. As we've seen throughout this session, cell painting can evolve as a powerful tool in phenotypic screening. With increasing adoption and technological improvements, its role in early stage research will grow significantly in the coming years. We've demonstrated that a wide range of cell types, including those with challenging morphologies, can be successfully imaged and analysed. And this opens the door to more biologically diverse and disease-relevant models, bringing us closer to in vivo physiology and allowing us to study responses in a controlled, reproducible way. And this is especially valuable for understanding mechanisms of action and identifying subtle phenotypic effects. Ultimately, expanding the cell painting toolkit enhances our ability to uncover mechanisms of action, predict compound toxicity, and identify new uses for existing drugs. By integrating diverse and challenging cell types, we're making this technology more powerful, more predictive, and more translational than ever before. And with that, I'll finish the presentation there. I'd just like to thank a number of people. First, the excellent lab team at Concept, who have been integral to this work. Without them, it would not have been possible to get done, so thank you everyone. Thanks to both Justina and Daryl for providing expert scientific input throughout this process, which I'm incredibly grateful for. Thank you to our collaborators, both Cytochroma and Ignota, for allowing us to present some of the anonymized data. And finally, thank you to the wider Thermo Fisher team for their support throughout this process and with any troubleshooting questions we had. And finally, thank you everyone online for listening. Appreciate it.
All right. Thank you so much, Dr. Sullivan, for your informative presentation. We will now begin the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's go ahead and get started here. The first question that we have, let's see, it says, is there a method for coloring living cells and observing their changes in development?
So typically cell painting is used on fixed cells, not live cells. The dyes chosen need permeabilization to act, so the cells aren't viable afterwards, but there are a couple of options. You could use GFP tag proteins or acridine orange, and certain mitotracker dyes also don't require fixation. I know there's some literature regarding live cell painting, although it's not something I've really investigated at length. It's typically a trade-off with live versus fixed, in that you'll have less multiplexing capabilities and potentially you might get a lower resolution as well. So it can be done, but you've probably got a trade-off there.
Perfect. Thank you. This next question says, thank you for the presentation. My question is, how can we differentiate live and dead cells after using Painting Toolkit?
So the cell painting procedure itself doesn't specifically distinguish between live and dead cells, although you can use the information to infer viability. So things like nuclear fragmentation or cell shrinkage and blebbing and a collapse of the cytoskeletal structure can all give you an idea of potential cellular health. you might see that significant autofluorescence is a good idea of cell health as well. If you've got an image that is a small bright spot that's consistent across multiple channels, that's probably a good idea that you have a very unhappy cell phenotype there. But there is no specific, or there isn't a specific way to infer viability without changing some of the dyes because of the spectra that are involved.
All right, fantastic. This next one says, can you please say a bit more about the instrument itself? What objectives does it contain? Z-stacks? To which extent can you use it instead of the confocal microscope?
So the instrument itself, I can plug for Thermo Fisher. It's got a number of different objectives. The ones we have access to are between 10 and 40x, although it goes down to, I believe, 1.25 or 4x and up to 60. The Z stacking protocols that you can employ can be a number of different microns. So you can image hundreds of microns in sequence all the way through your planes. And you can do it with the confocal. You don't need to do an either or. So you can do it on the confocal itself, although it obviously does take a lot longer.
Perfect. This next one says, it seems that there is still heavy reliance on observing the phenotypic difference by eye after finding differences in the UMAP. Is there anything more specific that can be drawn out of the data beyond, quote, they are different under different conditions?
Yeah, so that's a fair question. And this is where building up the panel of reference compounds really comes into it, and having a large data set to back up the investigations that you're performing. All of the PCAs and the UMAPs have been generated from underlying data. So a lot of the images that I've shown in the presentations are the pseudo color composites, and they are essentially just pretty pictures. They can be altered to show whatever you want. The different colors and the relative intensities can be altered on them, but the actual analysis itself is done on the underlying single channel grayscale images, which were also present in certain slides. And they can't be altered. So the masking procedures that are performed on those are where the data output comes from. That'll remain consistent throughout experiments. And then when you've got a known mechanism of action of a reference compound, the idea should be that a similar phenotype would come and cluster together in the PCA analysis in a similar place to the unknown compounds and inform potential biological outcomes. And obviously, you'd require a large amount of data to do this. And I was just presenting a small proof of concept here. So I wasn't able to dive too much into that, but it is possible.
All right, wonderful. We are getting a lot of really great questions in here. The next one says, how do you distinguish between autofluorescence and valid fluorescence? From a flow cytometry background, this is done with fluorescence -1 controls.
Yes, so it is. It's not really amenable to FMO calibrations in cell painting. It's something that you will get a feel for. You can do individual channel scans that you can see. And as I've mentioned with the dead or dying cells, if you have a spec that is consistent in the same space across multiple channels, you'll probably have a good indication that that is going to be something that's not a true signal. It becomes something that you can quite easily catch by eye, as opposed to flow cytometry, where there's a bit more of a nuance to it. It's something that you can exclude from the data as well. So you can set thresholds in the masking software. So anything that is artificially high or extremely high above what you would consider normal, if you can then confirm it by selecting it in the software itself, you'll be able to see just where this very high signal is coming from, specific on the image, and decide yourself whether that is something that you consider to be a true signal or not, and exclude if it's not. So that's the best way to deal with it.
Perfect. It looks like we have time for a few more questions here. So this next one says, cell painting analysis is really complex and sometimes subjective to interpret. Do you have a fair criteria to determine if the analysis is good enough to resolve the different experimental conditions?
So again, it is going to be subjective. Although all of this is performed on different cell lines using the same set of compounds, using the same masking parameters that are captured during the same experiment, ultimately you will be the one that is setting the thresholds for those parameters. And this is where having the robust reference controls on the plates come in. You need to have those known phenotypes. You need to have those known mechanism of actions to really consistently say that you have got a true signal that you can identify. Having the vehicle control on every plate, regardless of what's being ran, having a known cytotoxic compound on every plate, and then having these known reference compounds that should have robust reproducible phenotypes give you the best chance to make sure that the data is as consistent as possible. But there is always going to be some subjectivity to it. So in that case, it's good to be able to do a validation against known literature.
All right. This next one that we have says, how do you determine which previously untested cell lines are viable candidates for cell painting studies, especially in early phase drug discovery?
So the ones that we selected was kind of a happy coincidence. It was ones we had access to and ones that were displaying different phenotypes. There wasn't a hard and fast criteria, and to be honest with you, I don't actually think there is a complete cell line panel that you could design. It's always going to be dependent on the question that's being asked. The main thing is if you can cover as broad a panel as possible to get as most out of the data that you can, that's going to give you the best chance to succeed.
All right. It looks like we have time for one more question here. So we'll go ahead and wrap up with this last one. It says, how many compounds and replicates have you tested per experiment and cell line? What pipeline do you use for data analysis? How can you see from the data without needing to look at the images what types of changes are compound inducing?
So that's several questions. So to break it down again, could you just give me the first part of that question again, please?
Sure. Yeah. It says, how many compounds and replicates have you tested per experiment and cell
Yes,
line?
of course. So we've tested up to 10 different compounds at a number of different concentrations, and the replicates varied between experiments, although we went as high as 10 replicates per plate. And it was very nice and consistent. One of the things that we were regularly doing was having an entire column of, say, vehicle or staurosporin treated controls, just to give that reproducibility. And in the plots that we were analyzing, you could see that they were highly reproducible because the clusters were very tightly together. Sorry, the next part of that.
No worries. The next part is, what pipeline do you use for data analysis?
So the data analysis itself is actually performed on the equipment itself. So it's run in something called HCS Studio, and that's where all the analysis is done. So you take representative images of some test wells prior to a full plate acquisition, apply the masking protocols on that, and then the software and the high content imager itself will be capturing the images and then analyzing the data at the same time. So as soon as the plate has finished imaging, the data is exportable as a CSV file or similar. And then we have access to a R-Shiny application that allows us to plug in some of the data into the PCA and the phenotypic analysis that we've been able to perform.
Okay, wonderful. And then the last part of that question said, how can you see from the data without needing to look at the images? What type of changes are compounds inducing?
So what type of changes? In theory, you can't, because this again comes to where you'd need the reference compounds that you'd have on the plates. Without looking at the images, you wouldn't know what type of changes are being induced unless you have those reference compounds that are consistent across cell lines. If they have the same effect, then a cluster that is appearing right next to, say, a staurosporin treated well might very well have a similar phenotype there that you can identify even without having to look at the images. The images are really a nice way of confirming what the underlying data is showing you. It's an added validation step, if you wish.
Thank you for that last answer there, Dr. Sullivan. And thank you very much for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Fisher Scientific, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. There were several questions that we did not have time to address. So those submitted that did not get answered and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Yep. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.
Thank you very much.